Okay, folks, good afternoon and welcome to another one of our TESS seminars. And as you all know, TESS is our Center for Tropical, Environmental, and Sustainability Science here at JCU, which is one of our most active centers in terms of particularly terrestrial work and freshwater work, but especially terrestrial ecosystems in the tropics is really what we're focusing on. It's an absolute pleasure today to have Professor Craig Moritz here. Craig's been in our orbit for such a long time. I first got to know Craig when he was at the University of Queensland. He had done his PhD at ANU, of course, one of Australia's top universities, as is UQ. He'd done some very interesting work on herps and on the genetics, and I won't try to get into the specifics of the molecular genetics, but I know one of the things he described there, which was published in Science as a doctoral student, was a realization that there was a two sexes within, I forgot the term for that, Craig will fill you in on that. So anyway, from UQ, he then went to the University of California at Berkeley, where he was the head of the Museum of Vertebrate Zoology, which is where I got my PhD many years ago. Craig was there for about a decade, maybe a little more than a decade. And then Berkeley, of course, is a really top university in the US and the world. And then went back to ANU as an Australian laureate, an ARC Laureate Fellow. And he's also now the head of the Department of Biological Sciences there. So Craig has been in high-level administrative positions as well as being a real international research leader for a long time now. So it's a real pleasure to have Craig here and to clean up my introduction and tell us all about his fascinating work in tropical and monsoonal Australia. Craig Moritz.
Thank you, Bill. Thanks, mate. So I want to start by thanking Bill and Tess for the invite. It's good to see you all here today, some old friends and some new friends. That's terrific. You are very like an undergraduate class, all sitting up the back or on Zoom. But please do ask questions as I go along. It could be a bit boring if it's just a monologue. And I'm grateful to Bill for not telling some of the stories he could have told about me, but I did tell him that I would have the last word, so he was remarkably restrained. So I'm a discovery junkie. I'm an evolutionary biologist. I'm interested in speciation. I'm interested in biogeography, patterns of variation. And I'm also interested in how we can translate that data to help drive or maximize effectiveness of conservation. But at heart, I'm a discovery junkie. I just love going out, collecting stuff, observing nature, observing animals in their natural habitats, and finding new stuff. And that really gives me a buzz. And so part of this talk is going to be about finding new stuff in areas that previously haven't been well studied. So I'm going to progress from, first of all, what a species is, because if I'm going to talk about discovering species, I better tell you what I think a species is and how we go about finding it. No two taxonomists will agree on what a species is, so I'll give you my version. And then I'm going to highlight some work that we did here in the Wet Tropics, starting off back in the days of the Rainforest CRC, when I first got to know Bill and several others of you here, and going on through collaborative work with Steve Williams and some of our joint students. And that's really about convincing myself that there do really exist in nature species that are indistinguishable morphologically, yet are good species. Good biological species, or whatever you want to call them. And that then underpins the extension of that work through the Monsoonal Tropics, which is what I did during the ARC Laureate, basically an excuse to get in troopy and drive all around Northern Australia collecting stuff and then analysing it. And then translation to taxonomy and conservation, highlighting work with Indigenous communities across the north. So that's kind of the plot. First, I have to acknowledge the people who actually do the work. I'm the pen pusher and occasionally a lizard catcher, although I'm pretty slow at that these days. On the right is the Monsoonal Tropics gang, taken at one of these Society of Herpetology meetings where everyone kind of lets go a bit. And then on the left, of course, is Steve Williams, who really guided my work here in the Wet Tropics. And still a good friend. So, what is a species? And this is really a slide I use in my second year lectures, trying to explain to students straight from an evolution textbook we sort of depict species as independently evolving lineages. So on the left, we have some ancestral form. Each of these little bars here is a population. They're exchanging genes among each other. And then something happens. They split into two sets of populations on independent evolutionary trajectories. So that's kind of the concept. And it all ties back to reproductive isolation, whether that be geographic, mating patterns, genetic incompatibilities, whatever it is that stops the exchange of genes between sets of populations. So that's kind of the concept, and taxonomists will argue black and blue about their favourite species concept, but most of them boil down to something like this. And you'll see, in particular, I focus on evidence for when lineages come together, is there substantial exchange of genes or not? And that kind of fits pretty much any species concept, biological species concept, an evolutionary concept like this, or whatever. So I put that in there just to explain where I'm coming from. Okay, now, what are these barriers and how do new species form? The process of speciation. I know this is a diverse audience, this is why I'm starting with this introductory material. And it comes in different flavours, and this is what I've shown you here is kind of a dichotomy, but there's a continuum in between. So the sexy end of speciation research is the so-called adaptive speciation. We have strong divergent selection based on ecology, on diet or habitat use, habitat specialisation, anything where there's strong divergent ecological selection. And this diagram shows an outcome of that. On the x-axis here, if you like, is time. This is sort of shown here as a phylogeny. Gee, this is hard to see. Shown here as a phylogeny. They end up with very distinct morphologies or ecological properties shown by different colours here. And this process can occur with ongoing gene flow if the selection is strong enough to drive the populations apart. So now, particularly with applications of genomics, lots of people are studying speciation with gene flow in which you require some form of divergence selection. The stuff we're seeing a lot of is really boring by comparison, but I think very prevalent. And that is where you get very long isolated populations, adapted to the same environment, but accumulating distinct mutations that when brought back together are incompatible. So they're adapting, it's ecological. So think of a rainforest critter south and north of Cairns, in different blocks of rainforest. They're not meeting up. They're adapted to their rainforest, but they're adapting to the same habitat. So it's like parallel selection, parallel mutation. Which of the mutations are different can cause genomic incompatibility. And these things are taxonomically invisible, because most taxonomy is based on morphology. It's only when you start looking at the genes that you see how common this stuff is. The other point here is that speciation takes a long, long time. So here, this can be very rapid. So short branches here depicted in this cartoon. This is very slow. This takes hundreds of thousands or millions of years because it requires separate mutations. The mutation itself is a very slow process. So this idea of protracted speciation. So what that means is that you have all sorts of little tips and branches and things. This is like a phylogeny of a clade of organisms. There's lots of tips here, just the ones at the top. This is very like the image in Darwin's Origin of the Species, the only image in the book. It looks just like this, where you've got divergent surviving populations, but back in history, going towards the bottom, you've got populations merging, so failed speciation events, you've got populations going extinct, and all we see is the survivors. So that means that when we look at sampling populations as well as species, we see this continuum of different levels of divergence, which is sort of the grey zone of speciation. Are you a species or are you not? And in an ideal world, we wouldn't even make that dichotomy. But to make taxonomy useful and put names on things, we have to dichotomize. We have to separate things. And the question is, how do we do that? All right. So this idea of cryptic species actually goes back to Dobzhansky and earlier. He called them sibling species in Drosophila, for example. And as people are doing this sort of large-scale sequencing now, two overarching patterns are emerging. This sort of morphologically or taxonomically cryptic diversity tends to be more common in the tropics than boreal zones in particular. This is data for birds. And it does vary by taxonomic group. It's quite common in reptiles, less common in some other groups. So I thought, well, let's look at tropical reptiles. Why not? Because it's fun. So the first part of the story is about the wet tropics. And we've known from a long time, from work even earlier than this, that Steve did with Richard Pearson, that there's a very strong imprint of climate history and different distributions of rainforest on the diversity in the rainforests around here. And so going back to Henry Nix, who was the first to really develop paleoclimate modelling or climatic modelling generally for species, he had an image very much like this in the Nixon Switzer book in 1991, I think it was. The purple areas here are the areas that are modelled to be continually suitable for higher elevation rainfalls. Not surprisingly, it's the mountaintops, right? And this is a surprise to some folks from overseas, because normally if you think of cooler temperatures, you expect stuff to move down the mountain. But of course, it's rainforest, so it's got to be wet. So it depended very much on the mist intercept and the cloud formation at high elevations. And this is borne out somewhat by or actually quite extensively by the plant fossil record for the wet tropics, which shows absence of rainforest in this area until about 8,000 years ago, when there was a rapid expansion of rainforests in the system in the cool, wet conditions of the early Holocene. So if you look at low dispersal species, lizards and frogs, there is a very strong imprint of stability, the persistence of rainforest, these darker areas, on overall species richness, independent of area. So clearly history matters here, and it shaped the broader patterns of species diversity across this system. And it's also very much shaped the genetic diversity and the evolution within species in the system. So on the right-hand side is a map similar to what I just showed you, and the arrows here are sort of cartoon versions of the predicted expansion of rainforests from the limiting conditions of the last glacial maximum. Remembering that was the last event in this 500,000 history of very large-scale climate fluctuations in the late Pleistocene. It's the one we know the most about. So this infers that there would have been major refugia here in the Bellnakur-Bartle Frere, the Great Divide Rim here, top of Mount Lewis and Mount Spurgeon, Thornton Peak a little bit around Finnegan, not much down south. And so the idea is that there was expansion from the central refuge to the south, and bidirectional expansion with populations that have been isolated for a long time now meeting together. And we see this in the genetics, and this was sort of the work I did with Steve. We went and sampled all over the place, a lot of fun. We'd sequenced this mitochondrial DNA. There's a phylogeny, you'll see a few of these. Short branches mean you're more closely related, deep branches, very different. And the biggest difference, sort of all going all the way around the loop here, is in this case around Lake Eacham and Lake Barine. So between these two major refugia on this map, it's sort of in here somewhere. And lots of other species have contact zones in this area like this. So you can ask the question then, what happens when these populations come back together? In other places, we have distinct populations here at Thornton and Windsor, down to about this area, Black Mountain itself, and then here's the Black Mountain corridor. So you've got populations here that are more closely related than either is to this one here. That sets up this wonderful laboratory, natural laboratory for studying divergence and speciation. How does this reproductive isolation evolve over time by comparing closely related and distantly related populations? So that's what Sonal Singhal, a PhD student at Berkeley, did. We went and sampled a number of independent hybrid zones, or contact zones that we mapped out. So this is 4 named species of lizards, but within them you've got closely related populations like I just showed you, and distantly related ones. Here's another pair in the red-throated skink, and some others here in the shade skinks. And we looked at the properties of the hybrid zones between them as a function of divergent history. So the gene sequencing, and assuming equal rates of molecular evolution, molecular clock that is, we could estimate splitting times of the two populations. Then ask what happens when these things meet in these natural hybrid zones. And the results were even better than we reasonably expected. The top right here, on the x-axis, we've got splitting time between these pairs. On the y-axis, we've got different measures of reproductive isolation. So on the left is a metric to do with genetic mixing in the center of the hybrid zone. On the right is just the percent of hybrids in those hybrid zones. And they both show a very strong relationship with time. And this is exactly what theory predicts. The longer two populations are isolated, the more genetic incompatibilities they develop, the more likely they are to be reproductively isolated when they come together. But there are still lots of hybrids. So a strict definition of the biological species concept would say, if you can form a hybrid, you're the same species. But people sort of move beyond that now. It's really about, can you exchange genes at a substantial rate between the two sets of populations or not? In terms of that original idea of species I showed you. And these graphs here are a little complicated, but I'll explain them. This is estimates from the hybrid zone studies of the position at which the genetic transitions occur geographically relative to the center, and how broad that change from one form to the other is, the cline width. So the stronger selection is, the narrower the cline, the selection against hybrids, and the more coincident clines are. And there's very strong relationship again here with divergence time. So the purple and blue here are the more divergent pairs, and they've got extremely narrow hybrid zones, and they're all in the genetic clines, and they're all in the same place. This is data from thousands of genes. And the green ones are slightly more divergent, less divergent, and the red one is these two very closely related things here. So again, a clear evidence that there's just no genetic exchange in these older pairs when they come together. And this finally convinced me that, these things really are separate species. They're just not exchanging genes on independent evolutionary trajectories. Yes?
So what you said you had required a substantial exchange of genetic material. How do you quantify it?
That's a good question. So it is a continuum. Some people will apply in a model selection, is there evidence for gene flow or not in model comparison. I think that's fairly crude. There are many sort of good species, good distinct ecological species that any taxonomist or ecologist would say, you're a separate species, which do have a history of genetic exchange, particularly genes that arise in one type, introgress across a hybrid zone, and then are adaptive or selected for in the other type. So that sort of genetic leakage actually underpins some adaptive radiations and so forth. Yet the things are regarded as separate species. So it's more than zero. If you think in sort of neutral terms, it'd be less than one migrant per generation, something like that, which is sufficient to stop neutral diffusion. But again, that's sort of forcing a dichotomy on what's really a continuum. Okay, so I think, for all intents and purposes, an evolution species concept, the purple and blue ones here, I would regard as separate species. They're just not exchanging genes, despite opportunity to do so. So one of these is up, if you go up the Gillies Road, just near the School for Field Studies, and go down that track, over 200 metres, you go from one type to the other type. And there's lizards everywhere, but no genetic exchange. So that's pretty cool. The other one's at Lake Breen. and Tinaroo. Okay, so Sonal then went on and we actually did the formal taxonomy. So in this case, these were the lizards that we compared. This was the red-throated lizards. These were the two that had no genetic exchange. And then this thing down here about Elliot was pretty distinct as well. So we grouped the green one as a separate species, the purple and red as another species. No, sorry, that's rhomboidalis. Purple is one species, and then these are the blue-throated. And again, similar process here, just where we have the hybrid zone data, there's no exchange, call them separate species. Even though morphologically they are identical. We measured everything in all sorts of multivariate statistics. You can't separate them. Now then, that left these mountaintop versions of the same species, Labrifornis robertsi, which are just above 1,000 metres. They don't form a hybrid zone because there's valleys between them. Yet some populations were more divergent from each other than we saw create a reproductive isolation in the other group. So if genetic incompatibilities are sort of clock-like, then we might as well go ahead and call them separate species because we predict that they would not be able to exchange genetic material if they come into contact. So we separated the two northern populations from the two southern ones, but didn't separate within either area. So that's sort of a preamble then to going on to the Monsoonal Tropics, which is a hoot of a place. It's really big. Right? So you start driving. And I love doing this as a troopie with our swags on top after a bit of rain somewhere, going through the Stokes Range here in Victoria River District. And from prior work, just assembling up the available mitochondrial data, it seemed that there was something going on. So per taxonomic species, there were at least two or more deep mitochondrial DNA lineages within those. Now, that's not enough to define them as separate species. You have to look at the whole genome, the whole nuclear genome to do it. So we thought, okay, let's go have a look. Let's see what's up there. And that's where the fun started. This is a nice little sign from Victoria River Downs. The people out there are just fantastic. It's really fun. So, of course, we're not the first to think of, let's look at Northern Australia. As I said, tropical biomes in general, of course, are very species-rich, and we expected to have very strong genetic diversity and divergences across the system. So, way back in 1986, Joel Crackcraft looked at bird distributions and proposed these biogeographic breaks between Cape York and the top end, and between the top end and the Kimberley, and others going south. And then since then, it sort of developed into this sort of plan with the Ord region as a barrier, the Gulf claypans, the Carpentarian Gap, and then other sort of large sandstone massifs, Kimberley Plateau, Arnhem Plateau, and the Selwyn Range, as logical sites for climatic refugia in the same way that we saw in the wet tropics. But of course, this is really flat. So I won't show you the results, but we've tried all this paleoclimate modelling across the system, and I don't believe any of it. The ability of the climate models to predict the monsoon in the current state is very poor. So I wouldn't trust historical models across the region very much. And it doesn't have the topography that we enjoy in the wet tropics. In the wet tropics, you can be off substantially in your estimates of rainfall and temperature and still get the same geographic pattern because of the topography. That's not the case up here. But I'd love to talk to anyone who's got more recent evidence on palynology or whatever. So, fascinating system. We trucked all over the place. I won't take you through that. And did a bunch of sequencing. Caught the animals and did a bunch of sequencing. And unlike previous work, when I was doing the sequencing in the wet tropics, it was laborious and painful because we had to Sanger sequence each gene independently. Really a pain. And now with these sequence capture technologies, we can sequence thousands of genes, if not whole genomes, in one hit. So the data suddenly become way more robust than anything we've ever had. So, and these same data sets can be used at big scale for phylogeny, such as shown on the right here for a clade of skinks. You can come down to comparative studies on in this case, 4 separate species, down to population genetics and looking at demographic history with the same data sets, which is rather satisfying, technically. So there's an example of the type of thing we saw. As expected, these things that are named as one species are not genetically homogeneous. It's a bit like the wet tropics again, but without such a clear overriding pattern. So each of these bars here is what's called a structure plot. It's an estimate of genetic mixing and the number of different genetic populations, in this case 4, one on an island here, then 1, 2, 3 across the range there. That's in Rostellus. AMAX down here I'll mention in a minute. It's got a very divergent population, as does another species sitting up on the islands off northern Arnhem Land, and additional structure across the system. So this is just sort of mapping out the diversity within species using these genomic methods. So are these things separate species or not? And we haven't had the luxury of doing detailed hybrid zones in this case, because there are so many species and access is tricky. But just for a thought experiment, I said, okay, let's look at the level of divergence where in the wet tropics we saw reproductive isolation in the same genus, and see how does that map out on these phylogenies. And so we've just got data on this one to show you, the Kali AMAX. So then we went and did a whole bunch of genome scanning of these 4 populations to see are they mixing when they come together, because again that's a very common species that we can sample throughout, and the answer is no. So again, that threshold that we developed for the wet tropics studies seems to work okay in this particular case in the same genus in a different area. There isn't a single magic number that you could apply to a dragon or a fish or a butterfly or other groups of lizards. But at least if you've internally calibrated it, it's a useful guide. So in fact, we followed up on the morphology of these, and there are some very subtle differences, and these should be described as separate species. Okay. We did the same sort of thing in little brown spotty geckos, a thing called Gohara, again, very common. If you look at any pile of rocks anywhere in the Monsoonal Tropics, it'll have, west of the Gulf, it'll have one of these things cruising around on it. The things at the top here were all regarded as this same species, Gohara, as were all of these. And then within this group, there were other things that were sufficiently distinct morphologically that the taxonomists had caught them and already named them. So this is not the pattern you expect for a single species. In general, single species are monophyletic or each other's closest relatives in one of these phylogenies. And so if you look at these things, they actually overlap geographically these or each other with no interbreeding. So good biological species, they're diagnosable morphologically once you know they're there. So they were all named as separate species. And I have to say, doing the taxonomy is actually really exhausting. So going out catching animals is fun. Doing the population genetics I love, and phylogenetics. Actually writing taxonomic revisions is painful. I'm looking at Matt here, right? But it brings together all this knowledge, all the ecological and geographic knowledge and population knowledge you have of species, and it's very low citation rates, and potentially the most valuable thing I've ever done, to actually go and name the species. So that's a plug for taxonomy. So these are the things with a new species. Down the bottom here, we've got this group here, which is continually distributed. And between these forms, they're shown here as being discrete. Do I have the slide? Yes, I do. When you do this sort of mixture analysis with SNPs now on these, we're seeing mixing between adjacent forms, particularly these and these. And we now know there's a hybrid zone here. So these things we would not regard as separate species, because we know in nature they are mixing fairly freely across their geographic boundaries. So that's the kind of approach we've been taking. Now, I'm also interested in how do you build up species diversity in a single place, and do you see ecological differences among sympatric species as you would expect. And indeed, we do. Once we knew what the pieces of the puzzle were, and we named the species and sorted them out, we saw really interesting evidence for size partitioning in local communities. You can get up to 6 different taxa together on the same big pile of rocks. And we spent ages in the field documenting exactly where we caught every gecko and how big it was and who it was, and came to the stunning realization that big geckos like big rocks and little geckos like little rocks. But at least we did the work to show it. And importantly, within sympatric combinations, we do see size partitioning. Now, that may not be related to the original speciation events, as ecological speciation. It could be character displacement subsequent or just size structured assortment of, at a community scale. But we couldn't see the picture until we sorted out who is who in terms of the species. So that was fun. All right, my favourite critter is this little guy, Heteronodia. This is the one in which I found the parthenogenetic population is built. Right? Never trust an ecologist with genetics. These are asexual triploid forms, but this is a story about the diploid sexual forms, which have staggering diversity across the Monsoonal Tropics in particular. In the rest of the continent, it goes right down to Perth and down to northern Victoria. It's just the pink form, the white dot, or the red dot. So there's 3 really widespread things, plus the clones. But then across the Monsoonal Tropics, there's about 30-odd different varieties of this thing. And it's largely habitat generalist, and it's really young. It's Pleistocene in age, compared to the Gahyra, which are Miocene in age. So what gives? So Stephen Zosiah, who some of you may know, he worked for Steve Williams for a long time and did his PhD with Conrad and Megan at Townsville, has been going out sampling all of these contact zones, like we did in the wet tropics. And we can do this with this species because it's ridiculously common and abundant. And Steve is an amazing field biologist. So he's done that now. And this data is slightly old, we've got more now, lots more. And the really stunning thing here is the lack of mixing when these things come together. And this is a habitat generalist, right? It's everywhere. If you drop a piece of tin in the arid zone and come back to it 2 years later and pick it up, there'll be one of these things underneath it. So again, these are these sort of mixing plots. Each bar here is an individual, and these are arranged across different parapatric contacts. And there's kind of nothing going on. This one is a mixing between 2 types. This is mixing. You can see the gradation of admixture proportions between them. But this one, this one, this one, this one, this one, there's no mixing where they're coming together. And that blew my mind. I really didn't expect to see this result. 30 species of Heteronodia. That is kind of shocking at the moment there's one. But it's a lizard this big that's distributed across the entire continent. So it stands to reason it's not just one thing. So he looked even further. This is the same data shown in a different way. These are called isolation by distance plots. If it looks like this, so this is geographic separation of samples on the x-axis and genetic distance on the y-axis. If it looks like this, then they're 2 separate species. If you account for geographic variation within, they're still really well separated, even where they're very close together. And these are the graphical version of this is geography now, latitude and longitude, and the colours here are the different genetic compositions. And you can see in the bottom 2, there's actually no hybrids at all, even though they're in very close proximity. And this one, there's sympatric, and there's no hybrids. So that's as good evidence as you can get for separate species. And the top one, by comparison, you've got a few hybrids and back crosses and mixing at that scale. So you'd call that one species. So that was a huge surprise to us. And there is some relationship here with divergence time. So again, on x-axis here, we've got genomic divergence, or think time. The y-axis is the average mixing in a 50-kilometre radius around the centre of the contact. And there's a couple of ones I pointed out as having evidence of mixing. And they're the most closely related things in the whole mess. And very quickly it drops down to zero, or effectively zero. This is a log plot of the same thing, log plot. So you can see a very quick drop-off in any genetic exchange. Much quicker than we saw in the skinks. Much, much quicker. So what gives? So in a couple of cases, it's clear that there is ecological divergence that's associated with a reproductive isolation. So although I say this thing is a habitat generalist, once you break it into the pieces, you start seeing some patterns. So this is right around Townsville. So Maggie Island and the rocks on Cape Cleveland, and up here in the Paluma Range, you've got I think this is a, tell when someone else gives me a slide because it's animated. They're actually different. They're living on rocks in those places. They've got little expanded toe pads. There's morphological features that says you're a rock dude versus a ground animal. But again, they're parapatric, if not sympatric, in these sites. There's no genetic exchange. The other feature is the stuff we can't perceive. In this case, pheromones. So this was the real highlight of Steve's PhD with Conrad and Megan, the discovery that when you look in contact zones, even ones with almost no morphological variation, these are just up behind Townsville, I think, or northwest Queensland and around Townsville. If you look at morphological variation, all lineages, yeah, this is up around Lawn Hill and Mount Isa. If you look at morphology, they're kind of overlapping, very hard to tell apart phenotypically. If you look at their smells, the male pheromones, they're well separated. So maybe, and we haven't proven this yet, maybe females are selecting males on male pheromones, and that's what's driving reproductive isolation. Maybe. Again, not proven, but that's the hypothesis. So, other stuff going on. All right, so on we go. Group of critters after critters. There's been a big study now done on skinks. And here we see some species being collapsed. So a lot of skink taxonomy morphologically was done on stripes and dots, back pattern, and minor features of scales. And when you look at them genetically, it's really clear that some of the named things have no substantive genetic difference at all from widespread things. So the ones with the crosses here are ones that, this is coming out of Roboski's group, are things that don't stack up when you do the genomic studies. So it's not all about endless splitting and splitting and splitting. In some cases, we're seeing good evidence for lumping species. And of course, the reason why stripes and dots appear and then disappear is actually a fascinating question. So I think about adaptation and predator avoidance and habitat use. Okay, so I guess what I've tried to show you here is, I think, pretty strong evidence. A lot of things that we think of as one species are not. They're multiple species in reality. And in some cases, the opposite is true. And a lot of this is sort of fitting in with this sort of view of cryptic speciation, such as we saw in the wet tropics system, but writ large across the monsoonal tropics. And a few cases where there's clear adaptive divergence, like in the rock-living heteronodia. Okay, so this is the time, I'll skip over that. So if you look at the bigger picture, we've done this in many more groups. We've turned up also in Sidomys mammals and rock wallabies and various other frogs, various other groups we've looked at. I think we've probably underestimated the true species diversity by several fold across the monsoonal tropics. And I guess that's no huge surprise, but it is telling and I think important to recognise. And we still have a lot of taxonomy to do. Like I said, it's hard. So the other feature of the monsoonal tropics, of course, is that it's largely Indigenous owned or managed. And that's a really important feature. And of course, the same is true in the wet tropics as it is in the monsoonal tropics. So progressively, a lot of Indigenous communities and the TOs have been declaring their lands as Indigenous protected areas to protect biocultural values. And it's incumbent on Western researchers such as myself to work with those communities to try and explore the biodiversity values of what we can contribute to their work. So one thing I particularly enjoyed is going out with Indigenous ranger groups. This particular example is China Wall, west of Lawn Hill, just across the border of Northern Territory. It's here. And of course, when you do this, you get a whole bunch of kids, as well as the elders and the ranger groups themselves. And actually just watching their dynamic as the elders teach the young kids. The younger kids come out and they're better at catching lizards than I am, so they keep coming, what's this? I've got this one. How's this one? And then teach them how to prep stuff and record things and so forth. Really, really fantastic. So I've done a bit of this. It can be difficult in some places, easier in other places. But I want to now just tell you how I think our science can help the traditional owners and Indigenous rangers understand from a science perspective what's on their country and perhaps add value, observe biodiversity value that can help with funding of the IPA programs and ranger groups. So I'll give you a bit of an example in the top end and in the Kimberley. So in the Kimberley, this is going back a few years ago now, we had a linkage grant with the Department of Conservation to try and map the diversity as we were seeing it across the Kimberley, and then ask how well did their plans for expanding protected areas in the Kimberley pick up this cryptic layers of diversity. Of course, as we go along with itself, we're also getting new species records for existing taxonomy as well. So at the time, they were talking about trying to get conservation linkages across Theta, for example, between these two parks, linking here through Charnley River, where Matt can tell you all about Charnley River, that's where he grew up, and all this. AWC are very active in this area, now with a bigger footprint that I'm showing here. This pond river down here. And later I'll mention a new national park being set along the Fitzroy down in the southern Kimberley. So that all existed in the Kimberley Science Conservation Strategy. And we were asked, how do we map our diversity onto this? So as an evolutionary biologist, I do this stuff. Catch animals, I sequence the crap out of them, build phylogenies, look at their population genetics and all that stuff. Occasionally get around to putting names on things, often not. So you have this mismatch of named species, cryptic species that have been named, and other stuff that you know should be named but has not been named. So it's like Rumsfeld's known knowns, known unknowns, unknown knowns, and all that stuff. And if I show a conservation manager or a TO this stuff, their eyes glaze over, they give me a little pat on the back and say, yes, that's nice, go away. If you can put it on a map, or if you can name things and define units, that's useful. If you can put it on a map, it's even more useful. And this is really important when the taxonomy is in flux, because you've got some old evolutionary lineages that don't have names. You've got some things that are named that are really, really short branches in a phylogeny. And maybe they're valid species, maybe not. Particularly island things are very prone to that, rapid morphological evolution, and so on. So working with Dan Rossauer, who'd already developed this idea of phyloendemism, the idea was to map out the diversity above and below the species level. So take a group like the skinks, get a phylogeny together, get your best estimate of the range of all the tips in that phylogeny. So discrete populations, nearly species, deep stuff that are surely species. Treat them all the same and measure the branch length and the geographic range. And then what you're trying to find is areas that have small range but genetically very divergent populations that for a given area capture the most evolutionary history. That's called phyloendemism. Now here's an example, a little velvet gecko. It's got 6 different things here. One out on Groot, one in East Arnhem Land around the bottom, and then 3 things around the Arnhem Plateau and east to Litchfield. So we've got a pretty good handle on their distributions. We used all sorts of probabilistic estimation of who's where and so on, based on sampling. So if you weight by how long the branch is, the phylogeny, and how small the distribution is, then Groot Island, not surprisingly, pops up here. Long branch, tiny distribution. it carries more weight, if you like. So then you can do that for genus after genus after genus after genus, and then just add it all up to identify sort of hotspots of unique diversity, whether things are named as species or not. So we've done this now for basically west of the Gulf. And some of the areas that light up are really obvious. So the Arnhem Plateau, well, duh. We already know that's a hotspot for plant diversity and so forth, as is the Mesic West Kimberley. We do get a few surprises. These islands off northeast Arnhem Land I talked about before, which are in the Martha Carl IPA, the Wessel Islands, have a number of undescribed species on them based on our metrics for lizards. And these karst limestones are devoting the so-called Go-Go Reef system down around Fitzroy Crossing. Again, Indigenous controlled lands, mostly, but some pastoral as well. And then the islands off the Kimberley were just off the scale as well. They had a lot of deep branch taxa that aren't named. So that's starting to get useful because we can put it on a map. And then we can ask, of these hotspots we've identified, how well are they represented in a protected area system? Or how are they represented in the Indigenous protected areas? Are there particular places that are of high value but not really emphasised as yet? And one of those was this karst limestone system, the Napier Range, down around Fitzroy Crossing, which at the time didn't have any described unique taxa to it. But after our work, it was really clear that there was several reptiles there found nowhere else and very old lineages that were otherwise sometimes related to the Mesic stuff up here in the West Kimberley, just persisting in that limestone range. So we did a bunch of mapping. And this is Dan Rossauer's work, sort of mapping the known areas of P. But note here, we actually have very few samples in the East Kimberley. So that's a kind of known unknown, if you like. We don't know what, because you can't map what you don't know. Okay, so we could do sums on what's in parks, what's in IPAs, what's in NGO reserves, and the level of capture of this, about half of it was actually captured in some sort of area managed for conservation. But then recently, Bridget Campbell and Emily Jane Ems and others, and I'm sort of connected with it, have gone back into this big sampling gap in East Arnhem Land, all Indigenous controlled country. So it's done very explicitly with strong engagement with the TOs and rangers. They pick the places, they pick the questions. We sort of add the science to it. We've got linguists working with us as well, all that stuff. And again, that's really improved our knowledge of the place. We've got lots of new, much improved species identifications because we're doing everything with genetics as well as morphology, range extension, new records, and interestingly, there were very consistent genetic breaks across species, north and south of Blue Mud Bay, which happens to be the separation between the Lanapoi and Seal IPAs. That's kind of convenient. So they've got their own genetic resources to manage distinct from the next Indigenous protected areas. These also happen to be different IPRA bioregions, so they're different landforms, if you like, north and south of Blue Mud Bay. My point here is that there's tremendous value in working with the Indigenous owners. So coming back to the Kimberley, again, this is a map of the evolutionary distinctiveness or phyloendemism across the system, slightly different colour scale this time. Again, emphasising the West Kimberley, the islands, and then the limestones running down through Fitzroy Crossing here. So Dan Rossauer used a conservation planning algorithm. Bob Pressey was here for many years. He was one of the architects of this systematic conservation planning algorithms and so forth. This is an algorithm, it's the one Hugh Possingham developed. I forgot the name of it. But it's been modified to capture representation of the phylogenetic tree itself, rather than just species as labels. Then asking which areas would have priority if you were to expand the reserve system as proposed by the Western Australian government, Or if you include all the areas that are protected, where do you get the most bang for your buck? And surprise, surprise, one of the top priority areas was around these limestones, around Fitzroy Crossing, a bit up around Theta, possibly because we sampled the crap out of it, because we had really good access to it. And also areas around Kununurra and down here, Argyle system. So this is sort of useful both for prioritising something like fire management, which you want to do across the whole landscape, as well as additional areas for protection. One of those was the Fitzroy Crossing area, which again is in this area with the limestones. I think this was a proposed national park in here somewhere. So unfortunately it didn't include these limestones. We argued that limestone should be included, but at least the pastoralists there and the Indigenous owners were very excited about the data and committed to protecting it. So I'll wrap up there. Voyage through my life as a geneticist. Points out as utterly unsurprising that taxonomic under-recognised diversity is rife across the tropics, particularly in these low dispersal organisms. And that I think this really does reflect this process of speciation. One of my colleagues just called it species just falling apart by geographic isolation, spreading out and then being isolated and falling apart without any obvious ecomorphological differences in most cases. We're finding new hotspots of diversity, and there's more to be found, right? This isn't the end of the story. It's just the start, really. And then I really do think this stuff, when you want to put it on a map and engage properly with landholders, agencies and TOs, can have some value. And I'll leave it there. Thank you. And I'm happy to take questions, of course. If Bill will let me.
Yeah, I monopolize the questions.
Yeah.
Okay, questions? And also online? Questions, anyone? And also online? Oh, go ahead.
Hi, Craig. That was a great talk. Thank you. It's Noel Priest here. I'm on the
cool.
Atherton Tablelands.
Yep.
I had a question about the conservation genetics side of things. A few years ago, 2015, Pat Woolley and Michael Westerman and others separated Dasyurus maculatus, the northern quoll, into 4 separate subspecies. And the reason I mention it is that the northern quoll in legislation is still considered as one species, and yet there are 4 very distinct lineages running from the Pilbara, well, separated at Pilbara, one in the Kimberley, one in the top end, and one in Cape York and northern Queensland. But in the proposals for conservation, there's been some suggestions about reintroduction, or so-called reintroduction, possibly even from Queensland to the Northern Territory, because the population in the Northern Territory crashed. Do you see any dangers in that sort of approach about reintroductions where distinct lineages, which have been separated by arid or flood barriers for, perhaps Pleistocene or even earlier, the risks that might occur in terms of losing a species or a subspecies? Can you comment on that, please?
Yeah, really good question, Noel. So this is the question of, do you mix, if you've got evidence for genetic decline or just population decline, is there gain to be had by mixing populations? And I was having a great conversation earlier about work here on the tropical bettong. I think, some caution is needed, but doing nothing is sometimes the worst choice. So if you can augment populations, there's demographic value in doing that if they're closely related, and genetic value in doing that. And that's being done a lot now for mammal conservation work with reintroductions for AWC and bush heritage and arid recovery and so forth. They've actually become very keen on mixing stuff. My take would be to try and mix within these evolutionary lineages, if at all possible. Because even if a population is just slightly distinct genetically and they've independently undergone inbreeding, then you'll still get a lot of value by mixing them, what's called genetic rescue. If you do mix things that are if you have no choice, if you've just got one population imagine you were down to one population of the tropical betel. The only thing left is at Davies Creek, I'm looking at you. And it's going to hell, and you know it's inbreeding, you've got the evidence for it, what do? I would mix it with the brush-tailed betel, which is its closest relative. Give it a go. Probably do it in captivity first, right? To make sure you don't get three-headed betons or something, or that they will die. But I think that there's been a very notable ground shift in the last 10 years towards the possibility of genetic mixing. Even if things are named as different subspecies. So in the burrowing betel in the west, they've mixed the Barrow Island populations with the Shark Bay populations. They had different subspecies names, and Barrow Islands were much smaller, I think. And they mixed them out in mature, and it's going great. There's no evidence for outbreeding depression, and the genetic mixing is working really well. So I think we have enough case studies now to include it as a strategy, but I prefer to mix things that are more closely related if you can. Okay?
Can I ask a question? Can you hear me?
Yes, it's Jason, is it?
Dennis.
Yeah.
Dennis Walls here. Look, I'm a bird guide, and one of the things that fascinates us, of course, is the splitting of bird species. It's kind of related a little bit to the last question as well from Noel. We have, of course, globally now about 11,000 species of birds as opposed to 9,000 not so long ago. And even in Australia, you alluded to it right now in relation to the split between the Northern Territory species and the Queensland species. We're getting a split from the Cape down to Cairns with the cryptic honeyeater and the graceful honeyeater. And of course, that could happen as well with the hornbill firebird and the helmeted firebird. My question is, we used to base it on DNA analysis, and it was usually 2 to 3% differentiation. We know that funding is a huge issue in relation to bird collapse, the paradox of species collapse when we are actually finding more species through splitting. And there's a bit of a political pressure, of course, sometimes not to have a split, because that means people are going to seek more funding in order to protect that species, which was previously a subspecies. What is the current state of play in Australia when it comes to the authorities having the power to say this is a new species, and therefore, of course, attract the funding associated with that?
Yeah, so, boy, that's a can of worms. But very good question. So, potentially we could, there is a tendency to splitting versus lumping, and sometimes that's driven by trying to get a particular conservation outcome, which I think is the wrong way to approach it, because it distorts the science. So this is why I try and set a very objective bar onto what we split and what we don't split, and sometimes we lump things. Now, but that's very much a decision for an individual taxonomist applying their own criteria to set of data, and you can give someone the same set of data. I probably see that with orchids. Show them the same evidence, and someone will say, look, that's 5 things, another person will say that's 1. So I think we do need some sort of arbitration in this, because species names matter so much for resource allocation and conservation. And we don't want to suck up all the money on the wrong stuff. So you can weight species by how much distinct evolutionary history they occupy. So the platypus is worth more than the bramble K. melames, as an example. But I think also there's been, I know the Mammal Society, I think the Birds Ornithological Union do this too, and the herpetologists certainly now have committees set up to review taxonomic decisions and make recommendations, or they come to a consensus view, which the Commonwealth government at least is using as the basis for the national species list. So there is some oversight of it. There are legitimate reasons to split versus lump between different people, because it's not written in stone in some sort of Moses type fashion of what a species is. It's often a matter of perception or what criteria you apply. I don't know if that's a useful answer, but a lot of people are thinking about this. But I think it's a mistake to split things just in order to protect a given population. There are other mechanisms in the existing legislation to do that. It's called important populations. So for example, greater gliders, you can protect the population at Eurobodalla because it's listed as a separate thing. Does that answer your question?
Yeah, look, thank you. I know it is a can of worms because we even talk now about splits in relation to bird calls, for example, with the cicada bird, which has got 3 distinct calls up and down the coast. And so there's even talk now of splitting those 3 in relation to criteria which previously were not considered to be splitting criteria, because of course it was always to do with the DNA, and the idea now that we have got such sophistication when it comes to finding out what that DNA through the genome and so on is, and then there are different groups as well across the world, that you might have a body in the UK which has a different approach to the one in the US and so on. And in the old days when I was growing up, we used to just have 2 large global bodies, as I recall. And now we've got multiple bodies making these determinations based sometimes on national interest.
Yes, I know Steve Garnett and Les Christidis have been arguing for a single unified approach, and good luck to them, is all I can say. Oh, sorry. Yes.
Okay, thanks for your talk, Craig. So I have a question. So basically your phylogenetic metric is biased towards your lizards, right? I was wondering how much you think...
The metric is general. You're talking about the phyloendemism thing.
Yes. Your analysis was based on your samples.
Oh, that's what I showed you. Yeah, that was all lizards.
Yes.
Yeah.
So my question is whether you think that could be expanded into other groups. Other groups have shown similar patterns, or it's just a unique thing for lizards?
I'd love to know, to answer your question. So in the wet tropics, for example, plants are much less differentiated across geography than are lizards and so forth. Birds are somewhere in between, as you might expect, or perhaps you wouldn't expect that. So certainly there's big differences among taxonomic groups, life histories and so forth, and how they respond to heterogeneity. I would love to see data sets like this for other groups of organisms across the north. There's some data for snails. Matt, how are you going with the plants? Yeah. So I think it's coming along, and so you could, higher dispersal rates, more ecological generalism should reduce the patterns compared to what we see in the lizards. The rock-dwelling frogs are much more strongly structured than are the floodplain frogs, again, as you would expect. So you've got to really look at this through an ecological lens and see what you would expect. But the binos gecko, the stuff I showed with all the crazy lack of integration across its distribution, was shocking, because this thing is just everywhere. It's a generalist. So something else is going on.
All righty, Craig. Thank you very much for a really interesting presentation. Very broad in its scope, covered lots of stuff there. So thanks very much, Craig, and we'll look forward to, it's beer o'clock, by the way. Join us outside if you like.
That sounds excellent.
Thanks very much.
Cheers.